All right. I want to welcome everybody here today, and it's really an honor and a pleasure for me to introduce Larry Zipursky today for the CTSI lecture. Larry was really one of my inspirations for coming to UCLA. He was my faculty mentor while I was a junior faculty, my chair in biological chemistry, and over the years has become not just a hugely admired colleague, but a really good friend and advisor. And I know I'm not alone in this, because I really feel that Larry has, one, he's trained so many amazing scientists who now run their own labs all over the globe. And I think that he's transformed the research of many of us sitting in this room, both by example and also by his generous willingness to really engage in and think about all of our science. Larry was born and raised in Canada. He did his undergraduate in chemistry at Oberlin, and then a Ph.D. at Albert Einstein with Jerry Hurwitz, studying DNA replication in E. coli, where he became a biochemist. And then he moved to Caltech for his postdoctoral fellowship with Seymour Benzer, studying neural development in Drosophila, and became a neuroscientist and a geneticist. And he came to UCLA to the Department of Biological Chemistry in 1985. He became an HHMI investigator in 1991, an honor he still continues to hold. He's an elected member of the National Academy of Sciences, of the American Academy of Arts and Sciences. He's honored by many awards, including the 2010 Alden Spencer Award and the 2015 Louisa Gross Horowitz Prize. So in addition to all this recognition, he really is truly a generous contributor to the community. He's served on selection committees for the Searle, the Sloan, the McKnight, Jane Coffinchilds, and now the Helen Hay Whitney Scholar Awards. And he currently serves on the NINDS Council. He was the chair of biological chemistry, and he now directs the neuroscience research theme. So Larry's research is aimed at uncovering, the logic of neural wiring in the brain. So he uses Drosophila as a model system to understand how neurons navigate to and make synapses with their target partner neurons. And does this in Drosophila, which has a clearly defined anatomy and genetically tractable system, so that he can test mutants that don't wire correctly and thereby get at the molecules that mediate guidance in synapse formation. So in trying to sort of encapsulate what I consider to be the real beauty of Larry's work, I couldn't help but think about the British romantic poet and prophet William Blake and his poem, The Auguries of Innocence, which begins to see a world in a grain of sand and a heaven in a wild flower, hold infinity in the palm of your hand and eternity in an hour. And so, why did I think about this? Because here's the human brain, here's the mouse, and there's the fly brain, a grain of sand. And yet, really, after one set of beautiful experiments after the other, experiments in which every detail matters, in which there's 360-degree views of every detail, he's uncovered not just the specifics of neural circuit wiring in the fly eye or the fly nose, but he's really revealed general truths that are principles that guide the formation of neural circuits throughout evolution. So just as one example, his lab has identified the family of DS-CAM adhesion molecules, which undergo really massively extensive splicing and to generate tens of thousands of isoforms, and through their homophilic binding, they accomplish the principle of self-recognition and self-avoidance, which has emerged as being fundamentally important and evolutionarily conserved in neural circuit formation. And then more recently, as I'm sure we'll hear about, his lab has uncovered another set of IgG adhesion molecules, the DPRs and DIPs, whose complementary patterns of expression and binding contribute to layer-specific patterns of synaptic connectivity. In this way, he's really beginning to get at this fundamental question of what guides synapse specificity in the formation of brains. So Larry, it's with pleasure that I introduce you and that I look forward to hearing your science. And I'll get you back on track here.
Thank you, Kelsey. That was a far too generous introduction and a very hard act to follow. My lab's been interested in how neural circuits are established during development, a problem that we've been working on now for many years. And in particular, we're interested in the cell recognition molecules that are on the surface of neurons that allow them to interact with one another during circuit assembly. Now, our first clues to how these circuits form during development really came nearly a century ago from the work of Ramon y Cajal. Who else? The father of neuroanatomy, working away about 100 years ago, described both in the vertebrate as well as the invertebrate nervous systems. See if I can use this here. I thought I could use this as well as Owen Whitty. That was what I said before. We'll see. He described these large bulbous structures at the leading edge of a developing axon that he referred to as growth cones. He likened these to battering rams and argued that these were sensory motor structures that responded to signals in the environment of the developing embryo and then translated these into changes in directed motility, culminating finally with the formation of connections between neurons, allowing them to communicate with one another. In addition to these axonal terminals, also describe these extensive dendritic processes extending from the cell body. Now, this process of connection formation to some extent has been simplified, as exemplified both in studies in vertebrates and invertebrates, insofar as this process takes place in a stepwise fashion. Growth cones at the leading edge of an axon navigate their way through the developing embryo using so-called guidepost cells that produce signals that allow these growth cones to find their way through the embryo, finally reaching their target area in a process we refer to as target recognition. And these growth cones then extend to the target, and within the target region, they must choose specific neurons with which to make synaptic connections. And I'll focus, after this introduction, largely on our attempts to understand the molecular basis of synaptic specificity. So considerable progress has been made in identifying signals and the receptors on growth cones that respond to these signals through biochemical studies in mammalian and vertebrate systems more generally, as well as through genetic studies both in flies and worms. And in a very, very general way, we can think of these molecules as contributing in three different ways to connection specificity, through combinatorial, graded, and diversity-based mechanisms. And I'll describe each of these in the next several slides. During the late 80s and early 90s, and through the 90s to about 2000, tremendous progress was made in identifying signals and their receptors on growth cones. And these were classified into four categories by Marc Tessier-Levine and Corey Goodman in a very influential review article they wrote in 1996, some 20 years ago. And they divided these signals into four classes. Two so-called long-range cues. These are cues that are released from cells and are proposed to act in a graded fashion, either to direct growth cones towards them or to repel them. The prototypical signal, netrin, can act either as an attractant or as a repellent, and the difference in response is due to the receptors expressed on these neurons as well as the intracellular signaling pathways that act downstream from them. There are two additional classes, short-range cues that can act in a contact repulsive manner or an attractant manner. These include the F ligands and semaphorins and ECM components such as tenasins in the repulsive category, and members of the immunoglobulin superfamily, cell adhesion molecules, calcium-dependent adhesion molecules of the cadherent family, and other ECM components. And as with the long-range cues, these signals can also act in a bifunctional way. The same cue can act either as a repellent or as an attractant, and they do so in large part due to their response in different developmental contexts. And finally, the second mechanism, gradients. And this is where different levels of the same molecules can play an important role in patterning neural circuits. And this is best exemplified by the effrins and F class of signaling molecules. Effrins as ligands in this context, and Fs as receptors, receptor tyrosine kinase on growth cones. These gradients of molecules play a crucial role in the establishment of topographic maps. And this is best characterized in the context of the visual system of vertebrates. This is one perhaps oversimplified version of mapping of retinal ganglion cell growth cones from the retina to the tectum. Graded expression of the ligand, effrin A2 and A5, high levels posteriorly and low levels anteriorly. And in the retina, retinal ganglion cells express different levels of the receptor that recognize effrins called Fs. These are receptor tyrosine kinases. They're expressed at high levels in the posterior and low levels anteriorly. And as a consequence, the growth cones expressing high levels are repelled. These are repellent molecules, are repelled, and thus they innervate preferentially the anterior part of the tectum. And conversely, growth cones that express very low levels of the receptor are capable of projecting to more posterior regions. The effrin B and FB class also plays an important role in this process, but they regulate patterning, this topographic patterning along the orthogonal axis. And this is a very complicated process. I've highly oversimplified it here. There are also other graded signals that play an important role in regulating this topographic map formation. And the third category is diversity. And these are large families of molecules with different ectodomains that have different recognition specificities, and they then signal downstream through a common signaling mechanism. dsCAM1 is a large family of cell recognition molecules that my lab has studied for some time in Drosophila. And a second very large class, the clustered protocadherins, have been studied in mammals, particularly in the mouse. And this work has been done, the initial work, including the discovery by Kimura and colleagues of a small family of these clustered protocadherins, and then Wu and Maniatis in 1999 identified the entire set of these clustered protocadherins. These genes generate diversity in different ways. dsCAM1 is subject to massive alternative splicing, so it's a single gene. And through alternative splicing, it gives rise to on the order of 38,000 different isoforms, 19,008 different ectodomains, and tethered to the membrane by 2 alternative transmembrane segments. And these ectodomains differ in 3 immunoglobulin domains of the 10 in this protein. And these differ in amino acid sequences from one another. The clustered protocadherins are organized in a different way. There are 3 clusters, the alpha, beta, and gamma clusters. Each of these alternative exons encodes an entire ectodomain. And for the alpha and gamma classes, these exons encoding a single ectodomain are spliced into a common set of 3' exons that encode a C-terminal intracellular signaling domain. Protocadherin betas each encode each of these exons encodes an entire beta isoform, including the ectodomain with its 6 different cadherin repeats, as well as the transmembrane domain and the intracellular signaling domain. These molecules are notable in that they encode remarkable homophilic binding specificity. They do so in different ways. ds-CAM proteins contain these three variable domains that I described on the previous slide. Each of these domains interacts with the same domain in a partner molecule on the opposing membrane. And these pair in an anti-parallel fashion through a single strand, a beta strand, both in Ig2 and Ig3, and through a common face of a beta sandwich for Ig7. These molecules are thought to be largely monomeric and flexible on the surface of opposing cells. If they have matching Ig2, 3s, and 7, they can then pair. We speculate they undergo this conformational change, giving rise to this twofold symmetric double S-shaped structure that's been characterized using X-ray crystallography by Mike Sawai and Woj Vojtrovic in a collaborative effort between my lab and David Eisenberg's. This conformational change is then argued to lead to activation of intracellular signaling, and this intracellular signaling leads to a response of dendrites in a characteristic way that I'll come back to, dendrites and axonal processes in a characteristic way that I'll come back to in a moment. Well, protocadherins are kind of wimpy. There are only 55 different ectodomains. Each of these show isoform-specific homophilic binding, but remarkably, these also show oligomeric-specific homophilic recognition. So oligomers will bind oligomers expressed on opposing cells. If they match at all of the at each of these monomers, they can then form they can then adhere to one another. By contrast, if two cells have only small differences in the combination of the protocadherin ectodomains they express, homophilic binding does not occur due to interference of the non-homologous or non-matching alternative ectodomains. These interactions occur in trans through these 4 N-terminal cadherin repeats. These are isoform-specific interactions. By contrast, the cis interface is isoform non-specific, and this allows for the formation of these heterooligomers. Now, this binding specificity would be consistent, of course, with these molecules mediating synaptic specificity, enormous potential for cell recognition. But certainly for ds-CAM1, and I think it's likely, although less compelling at the moment, that the protocadherins do not have this activity, but rather they play, as Kelsey mentioned, they play a crucial role in a process called self-avoidance and self-recognition, the tendency of processes of the same cell to recognize each other and avoid one another. ds-CAM1 isoforms, as well as protocadherin gamma isoforms, as well as the remaining beta and alpha, are expressed in a largely probabilistic way in individual neurons and endow each neuron with a unique identity. This identity allows their dendritic and axonal processes to discriminate between self and non-self. So in this example, for instance, we have molecules that match because they're on processes of the same cell. This leads to some type of intracellular signaling that is not well characterized either for the protocadherins or for ds-CAM1, downregulation of the receptor, activation of some type of cytoskeletal reorganization that leads to these processes extending away from one another. By contrast, different neurons express different isoforms which are largely different from one another. They fail to recognize one another, and that allows them to ignore each other, and their processes can then cross, unless, of course, these cells express other molecules that promote other interactions between them. Self-avoidance plays a crucial role in patterning the developing fruit fly brain, and increasing evidence argues that it plays an important role in regulating the patterning of neurites, both axons and dendrites, within the CNS of vertebrates. So if these proteins don't regulate synaptic specificity, what does? And what I'd like to focus on today is recent work in the lab in search of these synaptic specifiers. And we've approached this problem in the developing fly visual system, and we've done so largely for three reasons. First, there are a whole slew of great markers for different classes of neurons that we can use both to characterize the adult visual system as well as neurons during development. Second, this system is particularly well suited to genetic analysis. Genetic analysis at the level of single neurons. We can generate single mutant neurons in an otherwise wild-type background for either loss of function or gain of function experiments. And this type of genetic analysis provides the kind of resolution that one needs to get at the question of synaptic specificity. And finally, we're particularly fortunate that an entire connectome has been described through serial EM reconstruction from the work at Genelia Farm Research Campus of HHMI. In their efforts to reconstruct the entire fly brain, they began with their proof of principle with our favorite part of the visual system, so we were tremendous beneficiaries of an enormous amount of work. So I'll begin with an introduction to the fly visual system and then our approach to getting at synaptic specificity. The visual system is modular. It comprises columns and layers. Columns process information from different points in space, and we can think of layers as processing different kinds of visual information. And what I want to do today is to focus on our efforts to get at the question of synaptic specificity in this structure, a medulla column. On the right-hand panel is an electron micrograph of a section through the third of 10 layers in a medulla column. It's an incredibly complex structure. I've colored in 7 neurons there, which we'll be discussing in a moment. But in addition to these 7, each of these columns, which is approximately 8 microns in diameter and about 60 microns in length, comprises the processes of on the order of greater than 100 different types of neurons. 8 of these columns have been serially reconstructed at Genelia, 1 using conventional TEM, and 7 additional ones in a separate animal were reconstructed using Fibs microscopy. And this is a summary of the Fibs data. 7 columns were reconstructed, 900 neurons were reconstructed, 53,500 presynaptic sites and 315,500 postsynaptic sites. And this discrepancy between the pre- and postsynaptic sites underscores the prevalence of so-called multiple contact synapses in the fly visual system, synapses that have a single presynaptic release site that releases neurotransmitter onto multiple postsynaptic elements. So perhaps you can see that here. This is a presynaptic so-called T-bar, this electron-dense structure. And these terminals release neurotransmitter onto multiple postsynaptic elements. And here are 4 of them with very nicely delineated postsynaptic densities. So we set out then to identify synaptic determinants for these 7 neurons, R7, R8, and L1 through L5. And remember, all of these are actually in one of these columns. R7 and R8 are photoreceptor neurons, and they project directly to specific layers in the medulla. The L1 through L3 lamina monopolar neurons receive innervation from a different class of photoreceptor neurons that I haven't shown here. These 3 neurons, as well as the remaining 2 monopolar neurons, L4 and L5, also project in the medulla. These neurons have distinct morphologies in the medulla. They terminate in different layers, and they form characteristic patterns of synaptic connections. Remember, they're all in one layer, and that's, all in one column, and you can see that's organization from the serial EM reconstruction in the middle panel. Now, these neurons, remember, are joined by processes from 100 or so additional different neuronal cell types. There's synaptic targets. And some of these synaptic targets are shown on the right-hand panel. They fall into different classes, so-called medulla intrinsic neurons that arborize in different layers. Amacrine-like cells called distal medulla cells, and there are also proximal medulla cells as well that arborize within a layer and in different layers. And transmedullary neurons, these are like retinal ganglion cells in vertebrates where they make connections within the medulla and then project to deeper regions of the visual system, in this case, the lobule. Now, remarkably, each one of these classes of neurons is comprised of on the order of 20 to 30 different subclasses. Each of these subclasses arborize in different layers and elaborate different patterns of synaptic connectivity. So in this slide that no one should ever show, I'm trying to emphasize the complexity of these patterns. We're focusing on 7 neurons, these green neurons, L1 through L5, R7 and R8. And I've shown their arbor patterns here in this schematic in the middle, and 2 of their synaptic partners, one shown in red and one shown in blue. So up above here, in green are the neurons that we'll be discussing. Each one of these has multiple synaptic partners. So for instance, L2 forms synapses with, is presynaptic to 8 different neurons, and it is postsynaptic to an additional 6, and so forth for the remaining 6 different neurons. What's remarkable, of course, is that each one of these, if that weren't remarkable enough, each one of these neurons also has multiple pre- and postsynaptic sites. So it's a mess, incredibly complex, and foolishly, as a molecular biologist, you should always, I'm sure many of you have heard this Sidney Brenner quote, think big, work small. This is much too complicated, but bear with me. Hopefully we've made some progress in getting at what some of these molecules may be that specify these connections. Part of the challenge, of course, is coming up with ways of trying to fool the system and to circumvent this complexity. Hopefully we've done that. So genetically, we've approached this question in a number of different ways over the years. We've screened for behavioral mutants with the hopes that we would find behaviors that are disrupted by different disruption of different patterns of connections. We've also screened histologically. And while we've identified genes that regulate aspects of axon guidance and targeting, we've identified no genes that regulate synaptic specificity. And so with developing technology, we turn to using RNA sequencing to identify candidate molecules expressed on these cells and then to test their function genetically. So the approach we used was to purify R7, R8, and L1 through L5 neurons just prior to synapse formation using a set of markers that took some time to generate. We then isolated mRNA from these, converted them to cDNAs, and subjected them to Illumina-based sequencing. We then identified differentially expressed cell surface proteins, and we assessed their expression in vivo using mimic protein traps, and I'll come back to describe what these are in a moment. We then took advantage of a marvelous piece of work carried out by Chris Garcia and Engin Ozgen at Stanford, where they assessed the binding specificity of large numbers of Ig superfamily proteins and leucine-rich repeat proteins. And through this interactome, we were able to identify interacting proteins. We then determined the expression of these interacting proteins and sought to assess their function by genetic analysis. And I'll give you a progress report on 2 families of proteins that interact with each other that we've been studying over the past several years. So this is a control for the purity of the mRNA isolated from neurons purified by FACS. And here what I've done is I've compared the expression of specific transcription factors that we characterize by RNA sequencing. And here I've expressed their relative levels of expression via an RPKM value, and then compared this expression to of them with the immunocytochemistry of 7 transcription factors that are specific for different cell types. So for instance, this PDM3 is expressed with a very high RPKM value in L5 and very low levels in the other cells. And even for senseless, which is expressed at a relatively low level, 10 RPKM, a value of 10 RPKM, in R8, virtually undetectable in the remaining 7 cells. So the RNA that we purified from these preparations was highly pure, and this then set a criteria criteria for cell type specificity. Indeed, through other analysis, we identified genes that were expressed with an RPKM value of 5, that we had independent assessment that they were expressed in specific cell types. And we then used that as a threshold for expression in our differential gene expression analysis. This is a representation of the expression of genes encoding different cell surface and secreted proteins. So for a gene to make it onto this list, it has to have a minimum RPKM value of 5 in at least one of these 7 cells. Each column represents a different cell, and each row represents a different gene. 444 cell surface proteins satisfied this threshold criterion. And you can see at a glance that each of these neurons expresses a different combination of cell surface proteins. We found that cells express between 247 and 322 of the predicted cell surface and secreted proteins encoded by the genome. This is an enormous number. It's a quarter to a third of the potential cell surface molecules expressed by the genome, far more complex than we imagine. Between 49 and 168 differences were seen between any pairwise combination, with differential expression being defined as a minimum of a 5-fold difference between the 2 cells, and a relatively small number, between 5 and 14 proteins, were expressed in only one of the 7 neurons, again, where the difference of expression is minimally 5-fold. These proteins fall into a number of different superfamilies, including the immunoglobulin superfamily, the leucine-rich repeat superfamily, as well as those with EGF-containing repeats. And there's a large family of tetraspanins. And on this slide, what I've shown you is the differential expression, or representation of the differential expression of these immunoglobulin superfamily proteins. And again, you can see each column is a different cell and each row is a different gene. And you can see that each one of these cells expresses a different combination of these Ig superfamily proteins. Some of these, such as those listed on the left, are parts of subfamilies of this superfamily, and on the right, sort of rogue Ig proteins where there's just one of a particular type. In this slide, I've compared the candidates for synaptic specificity in L1 and L2, first showing you the synaptic maps, reminding you of these synaptic maps for L1 and L2. These neurons are remarkably similar to each other. They have virtually the same dendritic morphology in the lamina, where they are innervated by a particular class of photoreceptor neurons, and their patterns of synapses in the lamina are remarkably similar to one another. But their synapses in the medulla along their axonal processes are very different, and that's what's shown here, with about 5 postsynaptic or synaptic partners of L1, and about twice that number for L2. This shows the differential expression of cell surface and secreted genes in these 2 different cells. As you can see, most of the cell surface proteins are the same, and by the same I mean there's less than a 2-fold difference in their level of expression. But there were 53 different cell surface proteins that showed differential expression with a 5-fold difference in expression. And those of the immunoglobulin superfamily are shown here on the right, L1 enriched and L2 enriched, and you can see just by glancing at this, there are marked differences in the level of expression between these different cell types. We were particularly interested in this DPR family, 3 DPRs in L1, 5 DPRs in L2, And if we go back to this representation, the DPRs are here, and you can see that not only L1 and L2 are different, but so too for the remaining 5 different neurons. And so we focused our attention on this class of cell surface molecules. The first DPR identified was done so through genetic analysis by Craig Montell and his colleagues in a behavioral screen, with DPR standing for defective proboscis response. There are 20 additional paralogs of DPRs, and Chris Garcia and Engin Özkan and their colleagues in this large interactome screen they carried out identified another family of cell surface molecules that interact with these DPRs called DIPs. So there are 9 different DIPs, 21 different DPRs, and they show a complex set of interactions. Some of these are simple, such as DIP delta and DPR12. They are a monogamous pair, but the rest of these show promiscuous, much more promiscuous binding. For instance, DIP theta interacts with, looks like 5 or 6 different DPRs. A given DPR, such as DPR6, interacts with 4 different DIPs. And in addition, they identified a DIP they referred to as common DIP, or DIP-C, that's a leucine-rich repeat protein that binds to many of the DPRs and many of the DIPs. In a recent publication from Engin Özkan, he showed that the interaction between DPR that contains these 2 Ig domains and DIPs that contain 3, the interaction in trans between 2 different cells occurs between interactions of these N-terminal Ig domains. So we set out then to assess the expression independently of the DPRs to confirm, initially to confirm our RNA sequencing results. We resorted to this protein trap approach because we have been unable for many, many years to get any significant in situ hybridization in the tissue that we're studying. It's been a source of great frustration. But fortunately, Hugo Bellin and his colleagues came to the rescue by devising an incredibly powerful protein trap approach, and this work was carried out in collaboration with Hugo and his colleagues at Baylor. So they took advantage of a transposable element called a Minos element. These Minos elements hop around the genome, and they tend to hop into introns, which is particularly convenient. And Hugo and his colleagues modified these Minoses to make mimics. And mimics are transposable elements that are very easy to manipulate. So they have these transposon arms, a left and right, and then just inside they've inserted 2 recombination sites that are recognized by a bacterial recombinase called ATP sites. And this is a cassette, and we can easily exchange this cassette using 5C31 mediated exchange with pieces of DNA that are flanked by ATP sites. And so literally thousands of lines have been established that now in which GFP has been inserted into these modified transposons, and these function as protein traps. And remarkably, the vast majority of these protein traps are expressed in cells, and even more remarkably, many of them retain some biological activity. We were fortunate to have access to 10 different traps, or traps for 10 different DPRs, and these largely showed the same patterns of expression that we had seen by RNA sequencing, and I'll show you 3 examples here. So DPR15 GFP is strongly expressed in L1 neurons and weakly in L3. DPR13 strongly in L2, weakly in L4, consistent with what we'd seen from the RNA sequencing data. And similarly, L3 is remarkably, DPR17 GFP is remarkably L3 specific as it is in the RNA sequencing. This summarizes expression just before the onset of synapse formation. And you can see that each one of these cells expresses a distinct combination of DPRs. this DPR expression is to some extent dynamic. So at this stage, we can see that L1 and L3 express DPR15, for instance, and DPR2 is not expressed by any of these cells. If we go to 72% of pupil development, which is a period where many synapses are being made, we can see we lose DPR15 in L1 and L3, and we now see the appearance of DPR2 in L1. Some of these are stable, such as DPR12 is on in L2 and L4, both early and late, but many of these are quite dynamic. Now, remarkably, while there's dynamic expression of DPR in these cells, we saw very little expression of the DIPs, their binding partners. The DIPs, by contrast, are expressed in the target region. And this shows two different DIP GFP fusions, DIP alpha GFP and DIP eta GFP. The cell bodies of these medulla neurons, these target neurons, are packed into this, where is this here? Are packed into this cortical region. There are about 40,000 different cell bodies of, again, remember, 100 different neuronal cell types. And not in any particular pattern, they're not arranged in any particular pattern. These processes then extend, these cells then extend processes into the medulla neuropil where they arborize in particular layers. And you can see that alpha and eta show this layered specific expression, but these patterns, though overlapping, are clearly different from one another. We were fortunate to have 8 traps of the 9. Data for 6 of them are shown here. All of them are expressed in this layer-specific way. And I want to emphasize that we've been looking for these kind of patterns for a really, really long time. And I just assumed they didn't exist. So this was quite a remarkable finding for us, that indeed there are families of molecules that are expressed in this way. Now, remember, each of these columns contains more than 100 different cell types, neuronal cell types. So just by virtue of the arborization patterns, we can't really tell, or I should say these layer-specific patterns, we can't really tell what neurons are expressing these different DIPs. And so we turned to a technology that I'll come to in a moment, called MCFO, and I'll define it then in the next slide. But it's critically dependent on us being able to now take these mimics and convert them from a protein trap to a transcriptional trap. So instead of having GFP, we've introduced then GAL4. And GAL4 then gives us an entry, and I should say these GAL4s are expressed in the same pattern as the protein traps. So we combine this GAL4 with a method devised by Alyosha Nurin at Genelia that he calls multi-cell Flipout, or MCFO. So we construct flies that have the following set of genes. They have a GAL4 insertion into a DIP gene, and the GAL4 is expressed in the same pattern as the DIP. We then have a transgene that encodes a recombinase, flip recombinase, that we can control by limited heat shock. So a little bit of heat shock, short period of heat shock gives a very brief pulse of flip expression, and then longer, more flip recombinase. And these flies now contain 3 different transgenes that are inactive transgenes. They've been rendered inactive by insertion of recombination sites that flank them. And within the guts of this insertion is a transcriptional stop signal. So nothing is expressed from these GAL4 binding sites, these UAS. These 3 genes are related to one another. They're derivatives of GFP, in which, through work at Genelia, a whole slew of epitopes have been inserted, which gives a particularly good target for antibodies, but also inactivates the endogenous fluorescence. So, in these flies that contain these different genetic constructs, no target genes are expressed. because each is interrupted by this so-called interruption cassette. So by giving a brief pulse of heat shock, you can activate flip, and you can get different combinations. In some cells, you'll get just a single. In others, a double. In some, 3 different will be expressed, and so you can get 7 different colors. And this is a way of being able to resolve from a complex pattern individual neuronal morphologies, and that's shown here. So this is a dip eta gal 4 on the left, and although we can tell that it's layer specific, we don't know what cells are expressing it. And so here what one can do is to use this MCFO with a dip epsilon gal 4, and with a very brief heat shock, we can get a small number of cells that are labeled, and we can now determine the identity of these cells, not by the genes that they express, because unlike the lamina where we have nice transcription factor markers for each different cell type, we have very few markers in the medulla, but we can determine the identity based on its morphology. So this shows a so-called TM4 neuron with its cell body here, a very characteristic branching pattern in the medulla, and terminating in a specific layer in this deeper neuropil called the lobula. So that's TM4. This is a DM3 neuron that arborizes only in a specific layer within the outer part of the medulla, and so on, for instance, for DM18. And we can do this for other dips, such as dip eta, shown here. This is a related, another type of transmedullary neuron, TM3, which arborizes in a pattern that's different from TM4 that I showed you on the previous slide. It, too, also projects to this lobular region where it terminates. This one, I've got to follow it here, terminates here. This is another transmedullary neuron called TM2, a similar but a different arborization pattern in the medulla, and you can see that it terminates here in this different layer. So this is a way that we can sort through all of these dips and determine the pattern, the cells that express different dips. This is a particularly neat one here, this TMY3 neuron. It's like TM neurons, except when it gets down to this part of its trajectory, the axon splits, and it sends one terminal to the lobulin and the other to the lobular plate. We can also assess matching of dip expression with cell types by guessing, and this is what we did initially. So for instance, dip alpha trap is shown here, and based on this morphology of these cells and the pattern that we see of layered pattern here, we guessed that DM4 would express this dip. We were able to then cross a DM4 marker that we have, a transgene marker, with a dip alpha containing fly, and we can then look for colocalization. You can see that a subclass of the dip alpha cells includes DM4. Okay, so here's just a very brief summary of dip expression in the medulla, and this is still work in progress. The dips are expressed in a layer-specific fashion. Each medulla neuron expresses a single dip. As many as 20 different cell types can express the same dip. One exception is dip eta and theta that have been found to be expressed in some of the same cells. And these two dips, eta and theta, have the same DPR binding specificity. So this perhaps is the exception that proves the rule. And these are all expressed during synapse formation, and expression is maintained into the adult. So returning then back to L1 and L2 and synaptic partners, we can begin to match the expression of these interacting proteins in different synaptic partners. So let's look here at L1. I showed you a morphology with TM3 with dip eta, and you can see that dip eta and theta are expressed in TM3. L1 expresses a DPR, DPR2 and 3, both of which specifically interact with this dip. Look at L2 here, and I showed you that dip epsilon is expressed in TM4. L2 expresses DPR6 and 13, and DPR6 and 13 bind to dip epsilon in vitro. most of the pairs, not all of the pairs, but remember this work is still in progress, so I suspect other pairs will also express different DIPs and DPRs that interact with each other. So the big question, of course, is whether these actually have any role in regulating the interaction between these cells. And to address this issue, we've begun to make mutations in the DIPs and DPRs using CRISPR technology. Amazing, I got through this talk without saying CRISPR to right near the end. So we use CRISPR technology, which, like in every field of genetics, has revolutionized how we do it. It's wonderful to be able to design your null mutations and generate them. It's very effective. Indeed, you just send your sequences off to a company and they do it for you. So we've begun to knock out the DIPs and first and some of the DPRs. And here I'll show you some data on DIP alpha and DPR6 and DPR10 double mutants. Both DPR6 and DPR10 interact with DIP alpha. And just to remind you then, DM1 expresses DIP alpha, and its synaptic partner L2 expresses DPR6, and two different DPRs in L5 that interact with DM1, which is a synaptic partner. And L3 expresses DPR6 and DPR10, and L3 makes synapses with two DMs that express DIP alpha, DM4, and DM12. And what we've seen in our early analysis of these loss of function mutations is we see cell loss of the DIP expressing cell. So in this case, DM1, we see a 26% loss in DIP alpha, a similar loss in its ligand or its receptor, whichever its partner, DPR6, DPR10. And for DM4, we see a 44% loss and with the DIP and a 26% loss with the DPR, and a very modest effect on DM12 seen in DIP alpha. We've not looked at the DPR6, DPR10 combination yet. I want to emphasize we've also seen this with another pair, DM8, and its synaptic partner, R7. R7 expresses DPR11. DM8 expresses DIP gamma. We looked at null mutations in both of these, and they give rise to a very similar phenotype with a marked loss of cells. So selective expression of DIP and DPR proteins was observed in RNA-seq and mimic-based protein expression analyses. DIPs and DPRs are expressed on synaptic partner, and we see a loss of DPR-DIP pairs. Loss of DPR and DIP pairs through mutagenesis leads to a loss of the DIP-expressing neurons. So this loss could be a consequence of a number of processes, and we're really very much at the beginning of this developmental analysis. This could reflect a role for DIPs and DPRs in regulating the development of these cells or their differentiation. Alternatively, this interaction could act to provide a trophic factor to promote the survival of DIP-expressing cells. Or perhaps these proteins play a direct role in synapse formation, and in the absence of normal synapse formation, as a consequence, these cells may die. And certainly there are precedents for this in a variety of mammalian systems. So it's too early to say what they're doing. I'm thrilled that they're actually doing something. And I'm sure they're probably doing something that we don't anticipate. So I'm looking forward to uncovering this. I just want to, in the last few minutes, put this work into the broader context of what we know about assembly of columns, and that come from, and particularly focusing on this layer 3, which is where DM4 and L3 neurons are, and where this DPR610 DIP alpha interaction takes place. So this summarizes the work from a number of labs over the past 15 or 20 years. I've focused it exclusively here on the targeting to this M3 layer. So assembly of these columns occurs through a cascade of intracellular signaling molecules, and I'll describe that for the assembly of M3. So R8 neurons are the first neurons to grow in from the retina, and they initially target to this superficial region of the medulla, and they sit there, and they can sit there for days. They're followed in by R1 through 6 neurons, and these neurons release hedgehog from their growth cones. And hedgehog, in turn, then drives these lamina precursor cells through their final cell division. These growth cones then release EGF. EGF then induces the differentiation of the 5 lamina neurons, L1 through L5. Here we're focusing on L3. L3 then differentiates. It sends its axon past its target in M3, and it's prevented from projecting more deeply through its interaction between SEMA1A on the growth cone of L3 and plexin A on the terminals of differentiating medulla tangential neurons. And they also interact, and L3 terminals also interact through homophilic interaction mediated by N-cadherin with a variety of different neurons within this region of the medulla. If we remove both CAD-N and semaphore 1A, these guys just keep going to deeper regions of the brain. In a subsequent step, L3 then releases a ligand called JEB, and JEB interacts with the evolutionarily conserved ALK receptor tyrosine kinase, and this is a survival signal. It's essential for the survival of L3. If ALK or JEB is deleted, these neurons degenerate, and interestingly, the degeneration begins right here at their dendrites, and then the cell is lost. This terminal of L3 then reorganizes in a process that involves SEMA1A. It's unclear if it involves N-cadherin, and assumes its position within the M3 layer. And at this point, it begins to produce netrin. And netrin plays a crucial role in mediating the adhesive interaction between the terminal of an R8 growth cone and the M3 layer. And then in a subsequent step that I've described today, L3 utilizes DPR6, DPR10, DIP alpha interactions to promote, in some way, the development of DM4 neurons. And we know from some very recent experiments that if you delete L3 neurons, these DIP alpha terminals remain at this lower level. So presumably there's some other type of interaction that's important here that precedes the DPR6, DPR10 interaction. And we know that that interaction is not from netrin, but must be from some other signal that's being produced by these neurons. So I just want to remind you again that this is clearly just the tip of the iceberg. There are, again, 100 different neuronal cell types organized in these remarkably precise circuits. So there's going to be enough to keep us going for a little bit longer to identify the kinds of molecular interactions that specify these patterns. And of course, what we're really looking for, we don't really want to piece together every step. What we're really looking for are some principles by which these connections are established, the logic underlying the formation of the circuits. And we're hopeful that this approach will begin to unravel some of those principles. So let me stop there and thank a very talented group of people who did the work that I described today. The RNA sequencing work was initiated in my lab by a former postdoc, now in her own lab in Barcelona, Marta Mori, with some help from Jason McEwen, Matt Paco, and Wael Tadros. Most of what I showed you today was done in a collaborative effort between Liming Tan and Kelvin Zhang, a bioinformatician, now working at Merck, and Liming Tan, a senior graduate student still trapped in my laboratory. And he's done a marvelous job in characterizing much of what I discussed with you today about vis-a-vis the DIPS and DPR studies. And he's been recently joined by Shua Zhu and Qi Xiao, two very talented postdoctoral fellows. And we've been incredibly fortunate over the years to have marvelous collaborators, and that's true too for this work on DIPS and DPRs, including Hugo Bellin and two fellows in his lab, Sonal and Peijing, as well as Kai Zin at Caltech and Shinya Takamura at Genelia. And we've been recipients of terrific reagents and advice from Alyosha Nern in particular, as well as from Barrett Pfeiffer and Jerry Rubin. And some of the markers that we use to purify these cells initially came from a large screen that Ulrike Heberlein carried out when she was a professor at UCSF. So I will stop there and take questions. Thank you.